Okay, let's dive right into the ultimate biological mystery, how a cell actually copies its information. Think about it for a second. Before a cell can divide into two new cells, it has to completely copy every single piece of its genetic information. That way, both of the new cells know exactly what they're supposed to do. So every time a cell divides, how does it make an accurate copy of its enormous DNA molecule? I really want you to imagine the sheer scale of this problem. There's no tiny literal photocopier inside your cells, right? It can't just take a snapshot of the DNA and print out a duplicate. We are talking about millions and millions of chemical letters that have to be duplicated with staggering accuracy to preserve all of that crucial biological information. So how does a totally mindless cell pull off such an incredible feat of logistics?
Well, it starts with unlocking the DNA code.
To figure this out, we have to look closely at DNA itself. DNA is made up of these tiny building blocks called nucleotides. Check out these four specific bases: adenine, thymine, guanine, and cytosine. Scientists usually just call them A, T, G, and C to keep it simple. You can think of these four bases as the foundational chemical alphabet of our genetic code. Everything that makes you biologically you is written out in an incredibly long sequence of just those four letters.
And this right here brilliantly illustrates the secret to replication: complementary base pairing. These letters don't just float around and sit together randomly. They have super strict chemical rules they have to follow. Adenine, or A, always pairs with thymine, T, and guanine, G, always pairs with cytosine, C. They fit together perfectly, kind of like two puzzle pieces. Because they are strictly matched like this, the two strands of the DNA molecule contain complementary information.
Let's actually see that chemical rule in action. Look at the original sequence at the top, A, G, T, C, C, A. Because of those strict pairing rules we just talked about, if you know one side, you automatically know the other. A pairs with T, G pairs with C, and so on, giving us our bottom row, T, C, A, G, G, T. This is absolutely crucial. Because of this one rule, an original DNA strand acts as a perfect template to predict and build a totally new complementary strand. This is the big aha moment that makes copying DNA entirely possible. But wait, we have a physical problem here. DNA is famously a twisted double helix, and it's locked tightly together. So how do we get inside to actually read this template? The two strands of the DNA double helix have to be physically separated before the copying can even begin. Enter an awesome enzyme called helicase. Helicase basically acts like a molecular zipper. As it moves along, it unwinds that DNA double helix and separates the two strands. Now, a really important note here. Helicase gently disrupts the chemical interactions holding the two strands together right down the middle. It does not break the sturdy outer backbone of the DNA molecule. By pulling them apart like this, it creates this Y-shaped opening that scientists call a replication fork. Okay, so we've got the strands separated, the template is ready and exposed, but how does the cell actually know what to put in that empty space? Now that the template strands are exposed in the replication fork, a molecular builder needs to step in, look at the exposed letters, and match up the correct puzzle pieces. Meet the builder, an enzyme called DNA polymerase. DNA polymerase moves right along the newly exposed template strand, grabbing free-floating complementary nucleotides and attaching them to the growing new DNA strand. And remember, it doesn't just randomly guess which piece to add. It is strictly following those A to T and G to C base pairing rules we saw earlier. It's just chemistry doing its thing. Okay, so helicase opens the DNA and polymerase follows right behind it, building the new strands. Sounds perfectly simple, right? Well, actually no. Why can't DNA polymerase simply copy both strands continuously at the same time? Here we hit a massive biological roadblock that the cell has to cleverly work around. We call it the directionality roadblock. The two original strands of DNA actually run in completely opposite directions, kind of like cars driving on two different sides of a highway. The problem? Our star builder, DNA polymerase, is like a car that can only drive forward in one specific direction, a one-way street that scientists call the 5' to 3' direction. Because the template strands are pointing in opposite directions, but our builder can only move one way, they simply cannot be copied in the exact same manner. This perfectly contrasts our two strands, creating this fascinating dynamic at the replication fork. One strand, called the leading strand, gets it super easy.
Because it happens to be facing the right way, DNA polymerase can just lock on and synthesize continuously, smoothly following the helicase straight into the fork as it unzips. But the other strand, the lagging strand, it's facing the wrong way. The DNA polymerase on this side is stuck working backward, away from the fork, meaning it has to build the new DNA in discontinuous, short little sections. It's like trying to pave a road while walking backward. So let's look at solving the lagging strand. Before we can solve it, we really have to ask, where does the new DNA actually start from? You might think DNA polymerase just grabs a letter out of thin air and starts a chain, but it can't. DNA polymerase is a fantastic builder, but it is totally incapable of starting a brand new strand from absolutely nothing. It always needs an existing starting block to attach that very first new letter onto. The cell solves this tricky puzzle using 3 brilliant steps. Step 1. A little helper enzyme called primase swoops in and lays down a short starting block called an RNA primer. Step 2. Now that there's a starting point, DNA polymerase jumps on and builds a short DNA segment backward, moving away from the fork. These short backward segments are famously called Okazaki fragments. But because the fork keeps unzipping further and further up, primase has to keep dropping new primers, and polymerase has to keep building new short Okazaki fragments. Finally, step 3. An enzyme called DNA ligase acts like molecular glue, coming through to join all those separate Okazaki fragments into one beautiful, continuous DNA strand. So now we have millions of bases being copied, unwound, built backward and forward, and glued together, which leads to a pretty terrifying question. What happens if it makes a mistake? Copying millions of letters at lightning speed isn't perfectly error-free. What if DNA polymerase accidentally puts a T where a C is supposed to go? Well, thankfully, DNA replication is highly accurate because DNA polymerase actually has incredible built-in proofreading abilities. It essentially acts like a molecular spell checker, double-checking its own work as it goes, catching and fixing the vast majority of wrong nucleotides immediately. And if it misses one, the cell has other repair mechanisms that come in behind it to help fix the spelling. But it's not 100% perfect. The occasional unrepaired changes that slip through become mutations. Now, these mutations can be entirely neutral, they can sometimes be harmful, or, fascinatingly, they can contribute to biological variation, which literally drives evolution over millions of years. And that brings us to the final result. The result of all these molecular machines working together is what scientists call semi-conservative replication. I know, it sounds like a big fancy vocabulary word, but let's break down what it actually means. When the cell finishes copying, you don't end up with one entirely old DNA molecule and one entirely new one. Instead, each resulting daughter DNA molecule contains exactly one strand from the original parent DNA molecule and one newly synthesized complementary strand. It basically conserves half of the original in each new copy. It's such an incredibly elegant way to ensure absolutely nothing gets lost. I want to take a step back from all the enzyme names for a second, because this is where it gets really mind-blowing. The cell isn't reading a book in the human sense. It doesn't understand the instructions it's copying. Instead, what we're looking at is a massive, elegant system of chemical rules. Things fit together simply because chemistry dictates they must. Complementary base pairing allows mindless chemical interactions to consistently and accurately produce profound biological information. You've basically just figured out how a system driven purely by chemistry solves the problem of preserving life's complex code. Which leaves us with a massive cliffhanger. We now know exactly how DNA can be copied so accurately. But think about it. Copying information isn't the same as actually using that information. If this DNA contains the literal instructions for making all the proteins that build your body and keep you alive, how does the cell actually read those instructions to build something? How do we get from a chemical code locked in DNA to a physical protein? Keep that provocative thought in your head, because that is exactly the mystery we'll be exploring in our next explainer.